Hello everyone, thank you for coming out to my dissertation events. My name is Raymond III, and today we'll be discussing hormetic abiotic stress in insects. I'm a PhD candidate here at New Mexico State University in the biology department, and I've been doing work here for 4.5 years now, and it's time for me to orally defend my dissertation. The work that I'll be showing you today is not all of my dissertation. It's about half, just because I don't have much time to talk about everything, but I do want to make sure I get a good enough story on what was done for you here. So to begin, I want to talk about research. My research began at Sam Houston State University, where I worked under Dr. Subaray Bushalai and Dr. Aaron Lin and the rest of my scientific colleagues, in which we were trying to understand human decomposition. This work was to help us understand the time since death, the significance of fly-borne bacteria, Ignationaria, on dipterans, on decomposition associated with human cadavers was my master's thesis, in which we did a lot of work on collecting the first 40 flies that arrived to the human cadaver. So me and Kelly did that. Also, were able to remove the different parts of the fly to understand the microbes that were present from the labellum, the tarsi, and the oocytes. This work with flies kept me wanting to understand flies and how maybe we can use those systems like such to help us further our understanding in science, to help us as humans. So I was able to come here to New Mexico State University, where I did work with Dr. Giancarlo Lopez Martinez, and I will be referring to him as GC in the talk. And I just want to note that this is going to be the light box shown here that I'll be discussing later on how we administered the different UVA to the pupae at the different shelf heights. Also, I want to point out that in this photo is some of the assay that we use to understand organism performance as far as flightability and even starvation. So my interest in research all began at Sam Houston and then brought me here, and these interests in entomology now led me to this interdisciplinary research in stress physiology. So I want to get started with showing you an outline of what we'll be talking about today. What is hormesis? Can we observe hormesis phenomena using abiotic stressors? My first aim is, can mild exposure to UVA radiation increase performance? And my second is, what genes are transcribed after exposure to anoxia and gamma radiation prior to adulthood? Let's get started on what is hormesis. So hormesis is a biphasic response, in which I discussed earlier this year in my literature review that we published. Me and GC published this, and it was entitled "A Dose of Experimental Stress Protects and Improves Animal Performance." This is one of the higher downloaded papers with the journal right now, and it's in reference to hormesis as a widely generalizable phenomena using homeostatic mechanisms following a stressor. You can see here that there are different animal models and organisms that were used in different studies that we reference, and aren't just insects or snakes, fish, or even mice. We also see plants and microbes. One of the things I want to point out here is the multiple stressors that were done with anoxia, and this exposure to anoxia hormetic conditioning is what was seen, and it enhanced the effects of the irradiation stress early in life in insects like the Caribbean fruit fly, Anastropha suspensia, in which there was definitely a change in how this organism performs. We also saw the same thing with the same anoxic treatments that enhance the understanding of SIT, or sterile insect technique, that allows us to kind of lower down those insect populations that are definitely invasive or even harmful to us as humans. So what is hormesis? Well, we know hormesis is a biphasic response to stress, where low levels of stress are protective and high levels are damaging. That these levels can be amount or time or a combination. So a nice multifarious approach to how we live every day, in which there's multiple stressors that occur from just us living and also stepping outside to the different environments that we live in. So hormesis is like exercise. Enough of it can be beneficial, but too much can be harmful, in which you see a lot of athletes being able to put in a lot of work and get good results when it comes to this exercise. And it's just enough to make sure that they are now beneficial above their nice, normal state of being. But then you see there's also athletes who take this too far, in a sense of taking in too much stress, too much exercise that is harmful. And you start seeing tearing in muscles and different aspects of aging that occur in athletes, and which you don't see many aging athletes because of this accumulation of damage that occurs. So for the past 100 years, for over 100 years actually, we see that there's been research on hormesis and this phenomena. So the effect of X-rays on the length of life of the tribulum confusum by Dr. Wheeler Davy was done in 1917, in which he looked at the flower beetle. And this flower beetle had a biphasic response phenomena that was shown, and it enhanced the consequences of this low dose of X-ray that increased the effect of the high dose of the detrimental shown in his study. And the same thing could be said in our own research that we published this year in the same review, in which me and GC were able to show this hormesis framework using flying adults as seen on the y-axis, and the actual stressor being X-ray exposure. So the increase in X-ray exposure shows this curve, in which there is a beneficial effect, helps for a maximum response above the no observed effect level. And this no observed effect level can also be thought of as the baseline, in which there is a homeostatic line or a base level in which an organism performs. And getting this increase in exposure, you see that this stress causes the organism to then be harmed below its normal baseline of exposure. So this increase in X-ray exposure shows this detrimental effect in which flying adults were shown to be less with the increase in X-ray exposure. But with the lower doses, there is a maximum response of the flightability that occurs after being stressed. With hormesis, why is this important? Why do I want to look at hormesis, and why are we even caring? Well, my work is focused on exploring the range of effects of environmental insults that organisms go through on a daily. This cross-tolerance to multiple stressors can have a protective mechanism, in which the importance of this is great in understanding how the different environmental stressors that we see possibly we can prepare ourselves for future stress. And using the electromagnetic spectrum shown here, my work is focused on this idea of how this affects organisms. Knowing that the shorter wavelengths of gamma radiation have this higher increase in photon energy, and also this increase in frequency as the waves get shorter. With the radio waves, you see the less of the photon energy with the shorter wavelength, and as this wave decreases in wavelength, we, like I said before, increases in frequency and this energy that accumulates in organisms and causes damage. We know stresses from the electromagnetic spectrum have antioxidant boosting benefits that are resulting in this cross tolerance to other stressors such as heat and desiccation. So with that being said, the importance of this is also dealing with all organisms on this planet, dealing with this climate change that we're seeing. The higher indexes, UV indexes that are occurring, higher temperatures, and the other things that happen in the process of our Earth warming. We also see that the UVC is coming through the ozone a lot more than it did before, and we're not sure how this is going to affect organisms in the future. But using this hormesis, we can understand maybe this dose response of how much UV you can get before it starts becoming detrimental. So now that we explored what hormesis is, let's talk about, can we observe hormesis phenomena using abiotic stressors? So we know organisms have evolved different defenses against stress. My work is framed by GC and Marcelo's work that shows organisms have natural defense mechanisms from environmental stressors and the result of oxidative damage. Both these individuals allowed us to see different studies done on organisms and understanding that there are natural defenses that occur, from the superoxide dismutase to the preparation in land snails to this oxidative stress. We know that oxidative aging and stress-related damage increases the amount of reactive oxygen species, ROS. And this ROS is produced in the mitochondria, and these studies show the mechanisms of pre-exposure of or preparation of stress that is still unknown, and my experiment is geared towards filling these knowledge gaps and pre-exposure to electromagnetic energy. So let's talk about UV radiation on this electromagnetic spectrum. Like I said before, we know that the UV is here later on in my talk, in which we did research on that as well. But here I want to start off with UV and understanding that UVA goes from 400 nanometers to 320 nanometers. The presence of UVA is shown in black lights, tanning beds, and are known to have the longer wavelength of the three, which shows that it has the less photon energy than UVC does. This UVA causes single and double-stranded DNA damage, shown in other essays, and this UVA is what I'll be administering to my flies and showing you in today's talk. But I must discuss that there's also other UVs like UVB, which ranges from 320 nanometers to 280 nanometers, that directly damages DNA and is a strong oxidative stressor. It reduces vitamin D production and has a medium wavelength between the three. And also UVC that goes from 280 nanometers to 180 nanometers. That is usually absorbed by the ozone. But like I said, with this increase in climate change and understanding that this UV is starting to penetrate through our ozone. So, UV radiation damages a cell. We know this. We know that UVA is an exogenous factor that generates reactive oxygen species, or ROS. We also know that UVB yields DNA double-stranded breaks that also involve ROS, the creation of these pyrimidine dimers, or molecular lesions on DNA. So it is free radicals, or ROS, that cause this UVA-related damage, which is my focus today. And a lot of our studies, including Benoit and GC studies, which they published in a textbook article, in which we know that there are mechanisms within the cell that are combating this stress naturally. So as we live in the mitochondria, we see that there's an increase in this ROS, or this hydroxyl radical, or the different oxygen radicals that occur, and the antioxidants needed to combat this stress. So we know that SOD, or superoxide dismutase, actually takes the hit as well, along with glutathione peroxidase within the cell. And this ROS, the reason why this is important is because it damages the cell membrane. Not only the cell membrane, but it also damages DNA and proteins and lipids within the cell. This damage that occurs allows for aging to occur, this accumulation of damage, and this understanding that damage itself is definitely going to be harmful to organisms. But we know that there is a natural defense for it, but at what points can this stress be beneficial? UV radiation and oxidative stress are related to this ROS, or the free radicals that are involved in creating this oxidative damage. This oxidative damage then damages the DNA, the proteins, lipids, which then increases the rate of aging. It's shown in many studies, and we also know that the macromolecular damage that is done increases cell proliferation, increased senescence, and neurodegenerative diseases occur with this increase in free radicals, or reactive oxygen species. The ecological effects of UV are still poorly understood, with low doses of UV having an increase in performance via an increase in antioxidant activity to combat this ROS, or this free radical damage that occurs. Long story short, if you're alive, you're experiencing stress anyway in the mitochondria, but at some point, getting this UV at a lower rate increases this antioxidant activity. Using this concept, I now want to ask, can mild exposure to UV radiation increase performance? To observe hormesis and this phenomena using UVA, we want to see if mild exposure to UV radiation helps lessen damage at cellular level. We know there's an increase in antioxidants, but is there a benefit in this increase? Is this method consistent and persistent? And what is the predictability of the UV intensity box? My hypothesis is that there is a hormetic range of exposure that positively affects performance. With this positive effect of performance, we're going to try to see this in Drosophila melanogaster as our model organism. My methods show in which we're wanting to apply this UVA to a lower developmental stage within the fly. So we stress the flies during a development right before adult emergence, or the ferret stage. So on the right here, in Drosophila melanogaster, we see that the larvae will eat and crawl up into a puparium, in which they now are starting to develop the different forms of eye. So the first stage here, we don't see many tissues that are formed that we can see through the puparium here. And as the insect grows and develops, we see that the 3 is where the pupa looks like once the adult has emerged from its puparium. I will be stressing the flies at the stage of at the 1 stage, right before we get to that developmental stage at 2. The implications for this is to understand that this stage is where it's non-mobile, and most stress tolerant phase of the development. Stress before this usually tends to have high mortality when stressing them prior to their puparium. And many studies have demonstrated the importance of DNA repair in the prevention of diseases such as cancer and other degenerative diseases. It is during the developmental stage prior to adult emergence that stress here can prevent emergence, shown in 3, and delay development. So why are we using Drosophila catanus, or the vinegar fruit fly? CS is a widely used strain of species in biology with strong genetic tools, and CS is a fly that originated from Canton, Ohio. And we can follow the flies their entire adult life. We can see the sexual dimorphism that occurs within the male and female, be able to sex those, and then see the process in which the flies grow. And we see that there are different stages and time periods in which the larvae are taking to get to their next stage. As you can see, there's a day in between each one of the larval instars. And then as it gets to the 3rd instar, it takes 2.5 to 3 days before it becomes a pupa. Half days of the pupa stage before it emerges and becomes an adult. This is that the fly has short developmental stages, and to develop a understanding of hormesis, we can now document this development and see aging and molecular effects due to stress with this organism. There's also a lot of genetic tools that allow us to knock down or overexpress different genes after experiments that can serve to help confirmation for hormesis in this phenomenon. The UVA treatments were administered at 0 minutes, being the control, 30 minutes, 45, and 60 minutes of UVA exposure, at each height of 7.62, 15.24, 22.86, and 30.48 centimeters at the shelf heights in the UVA box, as shown before, as I will discuss later. The original experiment started off with the 3 inches, 6 inches, 9 inches, and 12 inches, but in science we use the metric system. So I want to show these different shelf heights in centimeters for you. Here on the right, we see that all the experimental treatments were done in normal conditions, in which we're using a onset hobo, which monitors and calculates the data and gathers the data on the temperature and humidity that was present within the room of each one of these experiments. The organism essays that I will be discussing today that we ran to understand UVA treatments are emergence, flightability, starvation, activity, longevity, biochemical essays, and transcriptomics. These essays were done for us to further understand how UVA affects organismal performance. To administer the UV, which I'm sure a lot of you are wondering, the box that we created allowed us to change the different shelf heights. Use the UVA light, VWR lights, to administer the light to the pupae here shown at the bottom. This is the lowest of the shelf heights, which would be the 3-inch mark here, and the highest of the shelf in the experiments, which would be the 12 inches here. And as you can see, as the intensity increases, that the light will be closer, and as the intensity decreases, the light will go further away from the pupae. This UVA then is administered to the pupae in the puparium in their vials. The cotton in the plugs here are just to show that the vials are present, and the cotton plugs are not present while UVA is administered. And after the UVA is administered, one of the biggest things of these treatments that I wanted to do with this box, and why the box is very important, is because we wanted to administer UVA without this increase in temperature intensity that occurs in some studies. And here we were able to monitor and show that there is no significant difference between the different shelf heights as shelf height increased from the lower end of the shelf height at 7.62 centimeters or the 3-inch mark. We also see the same and the intensity of light. So we wanted to understand at what shelf height, what intensity is present. So UVA milliwatts per meter squared is shown here on the y-axis, and the shelf height on the x-axis showing you the curvature of this intensity in which we see. This principle is usually shown in other studies and mathematical principles, that there is a change in this magnitude that occurs as shelf height changes. And at the lower shelf height, you see this higher intensity as we increase the shelf. So the last two shelf heights here at the highest of the shelf heights were not done in all experiments due to the fact that we're looking for a hormetic or beneficial level of hormesis, and we want to see this change, and that has a magnitude difference from one another, so we can monitor and see if there's a hormetic effect, or understanding that see this phenomena that occurs within insects that's beneficial. So once again, temperatures were not high enough to affect insect survival, and the intensity was known throughout these experiments. Experiments were also done again in stable lab conditions using the VWR light, able to understand how much light is penetrating and coming into the room, and possibly affecting our assays here. So this VWR light was used at 365 nanometers in the experiments that I will show you here and the results. So after showing you the experimental conditions, now I want to show you the actual numbers. What happened here? So to do this, our first essay was an emergence essay, and we want to know, does UVA affect emergence? Here on the left side, we see that there is an emergence percentage on the y-axis, and the shelf height and the treatments in which the flies were administered with. As I said before, there's a control in which there was no UV, allowing us to get the baseline measurement of this form of hormesis. And then seeing at 30, 45, and 60 minutes, this curvature that occurs, and there's no significant difference shown in these different emergence patterns shown here at the different shelf heights. Insects, to make sure that we got the right percentage of data, so we're not looking at a smaller number and missing this hormetic framework that occurs in some studies. So overall, the total emergence was not affected by UVA. We see that to 3 inches to 12 inches that there is no significant difference at the different treatments shown below. Now that we know emergence was not affected by UVA, was flightability affected by UVA? And do insects fly? Is there a better flight pattern that happens when they emerge? of the greatest feats in evolution. So we were able to create a essay that allowed flies to be in the puparium here and fly out of the tunnel, the funnel, into the flying container where they're able to fly. The reason why this is black here is because flies fly towards the light, and this is an incentive for not only us to know that flies flew, but to also have and be able to calculate those flies that were marked as flown. So flies were counted after being outside of the funnel, placed inside the flightability chamber, and were marked as flown. As these flies emerged, we know that flies enjoy flight. I mean, that's what they do. And one of the biggest things with the data that I saw was that flightability was not significantly affected or significantly different from one another when UV treatments were administered at the different shelf heights, as shown below. And through all these different graphs, things to note is that on the y-axis here we have and the amount of flight after administering UVA. So the overall objective of understanding flight was done, in which we know that UVA at the doses that we administered does not affect flightability. At the different shelf heights, we did to 16 inches, shown here, with no significance in the amount of flight that occurs. We know that starvation is occurring in a lot of these organisms' lives in general when they emerge looking for food. And one of the biggest things about starvation is that when insects come out of the pupa, they already come out with all the nutrients they need. As far as when they're eating as a larvae, they gather up all the nutrients to come into the pupa, and as they emerge as adults, they're looking for food. So limiting the food source allows for a greater understanding of how stress affects the organism. In this essay, we did not want to dehydrate them, so we use this agar with no bacteria nutrients, which is 2% agar to water ratio, in which flies emerge in this container, and we're monitoring them for 72 hours to see the amount of mortality that occurs with the administration of the UVA at the different treatments. That data is shown here, as UVA does not affect starvation at the different shelf height of 22.86 or the 9-inch mark. We see that there is a significance in the starvation, in which the increase in this UV shows this increase in mortality that we discussed before, in which the higher intensity of the UVA radiation shows this mortality effect with the accumulated damage, in which we were looking at prior to. So Drosophila Population Monitor, or the DPM, activity levels of non-stressed flies at stressed adults at different ages. With the DPMs, or the Drosophila Population Monitors shown below, we're able to monitor flight activity in the vitals as they flew. Here at the top is where the cotton plug is, in the middle is their traveling, and the bottom one is where the eating or egg laying, or where the food is for flies to travel to get food, and also for the females to come and lay eggs. We see the same thing here in these, but with actual vials inside. So you can see here at the top, the cotton plug is used to make sure that the flies do not escape. We did essays looking at male versus females after 24 hours with control versus the treated groups, and we compared these treatments across the different treatment levels, in which the control of no UV going up from 30, 45, and 60 of UV administered. So one of the data graphs shown here shows us that UVA itself does not have an effect on the flies. But we know that flies themselves within our study, male versus female, have a significant difference amongst locations. We also see that there's a significant difference amongst gender, in which the location of travel activity was lower than the eating and egg laying. So they spent more time either resting or eating or egg laying And we also see that males found themselves more towards a cotton plug or resting than females. This fly activity was measured in movement, in which we were looking at the different stages and locations using the DPM. Now, does UVA affect flight performance? We know that in prior slides that the data shows there's no significance in emergence, flight ability, and we also see that there is some form of significance that goes on between the male and female flying between locations. But the effect of UVA treatment showed no significance in the flies themselves after fly activity was measured per minute per fly. So longevity studies allow us to see the lifespan and the longevity in which our organisms live. The vinegar fly lives 60 to 90 days throughout the USA and approximately 80 days in our lab because of the high desert and the low oxygen and humidity here in Las Cruces. We see that in these studies, we're able to have the flies emerge and then live on a weekly basis and monitor how they live throughout this approximate 80-day period. We remove the food and replace the food before the next generation of flies is able to emerge, so that we contain the flies and understand how many flies begin in the experiment. To do this, we do this weekly by taking this aspirator shown here and removing the plug. That plug is shown better in this image here. And we take out the flies from the bottom to count how many died per week. That data is shown here, in which we see that UVA has no effect on Drosophila melanogaster. At the 0 treatment, being the control, and the rest of the 30, 45, 60 minutes of UVA, we see that there is no significant difference amongst the organisms, especially when they get started. There's definitely this hormetic curvature that we see as time increases. And we also see that there is a percent survivorship here on the y-axis and the weeks in time. T-bars is something that we use to understand the damage done to the lipids, so lipid peroxidation. This oxidative lipid damage assay allows us to look at the diazobarbiturate acid, the reactive substance that occurs after oxidative stress. So malondialdehyde, or MDA, is the biomarker for lipid peroxidation of omega-3 and omega-6 fatty acids. So shown here in the image on the right, we can see that oxidative stress comes in and damages the cell membrane. As damage occurs, we see the MDA break off here, and we're able to quantify the MDA modified protein and monitor and accumulate, and that accumulated damage is shown as oxidative damage, in which I will show you the graph for here in a moment. I do want to note that lipid peroxidation affects protein synthesis in all tissues during aging. So the importance of lipids is why I chose this essay over the other ones to kind of present today, is because we definitely want to understand and know how lipids are affected in many organisms, but in flies, lipids are needed for reproduction, and shown here that the proteins are needed to protect and upregulate genes for protection and the defense of this the multiple environmental insults. Does UVA affect lipids? We see in the graphs below that at the different shelf heights, there is no significant difference amongst lipid damage that has occurred. We see on the y-axis, MDA per micromole of milligram of protein was measured for 0 to 60 minutes with no effect of lipids. But there is a form of variation, no significance in the sense of having more damage done by the different UVA treatments. We did biochemistry essays for quantifying performance. T-bars is the one I showed you here. But what and why we use these assays are important. We definitely want to understand the reactive oxygen species that are potent genotoxins that attack DNA, lipids, and proteins and generate oxidative products. These assays, including are able to monitor the damage done right? So DNA damage or oxidized guanine, and common assays like DNA breaks and repair can be measured, along with T-bars, looking at the different oxidative and lipid damage that happens within the cell. We also see that there's a lot of protein damage that can be monitored, and not monitored, but quantified and looking at the carbonylation of protein. We also see that there are defense mechanisms that can be quantified, and the total antioxidant capacity can be looked at to see what antioxidants are present, and if UVA is actually inducing this increase in antioxidant capacity. Also, enzymatic activity, or SOD, and catalase activity can be checked out, as well as the glutathione, in the sense of looking at the defenses. And going further past the damage and defense, we know that energy reserves are prevalent within organisms, including the Drosophila, in which carbohydrates versus glycogen can be measured, along with trehalose and lipids that are needed for flight and reproduction. Wasn't able to get to these different assays other than the T-bars and partial protein damage because of the departure of GC. But a lot of this information is important in our conclusion and understanding that exposure to prolonged UVA conditions early in life leads to no major effect in flight ability, emergence, starvation, and longevity in the experimental treatments. UVA radiation did not induce mortality under our experimental conditions. The accumulation of damage, which is likely found in reproduction, as in previous work studies, including Alyssa's work, our past master student in our lab, showed that there is an effect of this trade-off of living longer and not having an effective UV, or this anoxic radiation, or the stress that is in this hormetic framework and phenomenon that we discussed earlier in the presentation. Flies absorb 16 times more radiation at 3 inches than at 12 inches, showing that this increase in UVA should have shown some sort of damage or mortality. And we only see some form of variation in mortality, which is very important in saying that there were no negative effects that were measurable after exposure to UVA in the treatments that are shown here. So more mechanism studies that compare contextual factors and stress response would further our understanding of how the role of UVA hormesis plays in animals. So we just discussed my aim 1, and let's go into my second aim, my second question that I will be talking about today, and what genes are transcribed after exposure to anoxia and gamma radiation prior to adulthood. My data and my research, I want to find out the knowledge gaps. We want to fill these gaps in our understanding of how reactive oxygen species affects the cell, especially when it comes to biotic stressors. We know that biotic stressors are viruses, bacteria, fungi, semi-chemicals, and toxins, while abiotic stressors are droughts, xenobiotics, hypersalinity, thermal shock, radiation, and wounding. Knowing this information, we know biotic stressors enter the body and cause that internal response, while abiotic cause their internal response by attacking from and affecting the organism from the outside. And this ROS is increased, and it is through the mitochondria that the biotic and abiotic factors exert their damage. That is then sensed by the organism, which then leads to the transcription factors that are needed in defense. And these defense genes are activated. And as these defense genes are activated, is a coding that allows for the transcription of the protein needed to combat the different stressors that are occurring. We're also seeing the same process as the organism senses the stress, and protein kinase arise, and then transcription occurs, in which protein is then needed to combat this different stress. So increases or decreases to the mitochondrial function make the electron transport chain less effective and increase free radical production. And filling the knowledge gaps, the underlying mechanisms involved in defense are still unknown. Demonstration of beneficial effects of stress are lacking, and it is this excess of ROS that wreaks havoc in the cell. So I want to ask the question, what genes are transcribed after exposure to stress prior to adulthood? In the study we explored the mechanistic underpinnings of hormetic response of anoxia when combined with X-ray radiation, and its understanding of hormesis and this beneficial response of pre-exposure. And we want to look at this in a sense of multiple stressors. So normoxia itself is normal levels of oxygen. Gamma radiation is normal oxygen and ionizing radiation as an ionizing radiation dose. And we see that anoxia plus 80 gray of gamma radiation is the absence of oxygen and ionizing radiation dose. From here, the question that we asked now has to come with this hypothesis in which I formed, and knowing that hormesis is something that comes in beneficial. So my hypothesis is hormesis preconditioning has a strong protective response against anoxia and gamma radiation. Anoxia and gamma radiation on the transcriptome is very important in understanding how these genes are upregulated in the presence of the stress. So the libraries were constructed using a Illumina prep kit using 50 Drosophila of pupae. And we also used 2 micrograms of total RNA for each sample, and a total of 12 libraries were generated. The quality of the libraries were tested with a Agilent bioanalyzer, and sequencing was done using Illumina HiSeq 2000 system. And with the methods shown here, we did the treatment comparisons and understanding of normoxia versus anoxia and the other treatments as shown in the slide before, to compare these treatments to one another to see the response of genes and if they're upregulated or downregulated. Anoxia and gamma radiation on the transcriptome shows us in these volcano plots here of the differentially expressed genes with the different treatments as I showed in the previous slide, but now we're able to see these increases in gene products. So for normoxia and anoxia, we see that anoxia has the greater increase in gene products that occurs, with 283 compared to the 39 just living in normoxia. And the same can be said in the other graphs, in which we see that the stressor that is added to normoxia increases its upregulation more than just the normal levels of oxygen. I think the key thing to take in here is the multitude of genes that are upregulated, but in the presence of anoxia versus anoxia 80 gray in combination, we see this mass, and this is very interesting for me in the sense of this preconditioning that occurs, in which seeing anoxia combated with another stress and then nulling out the genes that occur in this uprising of genes is very informative in understanding that there is possibly a protective response in the presence of this anoxic stress. So seeing in the metagenes, we saw that several processes were affected across the conditions, with the organism's ability to respond to heat, the ATP binding that occurs within the cell, chaperone-mediated protein folding, protein processing in the endoplasmic reticulum, response to methotrexate, along with tissue development, cold acclimation, protein phosphorylation, carbohydrate biosynthetic processes, and also the response to hypoxia. And with the data that we got, there's a lot of genes in which I wish I was able to discuss here, but there was so many. I think one of the key things here is taking these genes and understanding their role that they play within being in this intense environment of being damaging the cell. So we know that genes MIR 2490 and genes heat shock protein 26 and heat shock chaperone 70-3 are upregulated during hormesis. And that type of defense could mitigate irradiation stress, as shown here in this masking effect that we see. And in other data, so I started off the presentation talking about the other hormetic experiments done in anoxia showing this preconditioning. And here in our study, we're able to show the same understanding that there are genes that come up and kind of mask the presence of this 80 gray or this gamma radiation. So to conclude, anoxia and X-ray radiation on the transcriptome, I want to discuss that anoxia radiation influences the expression of profiles of stress response genes. We also see that data provides further insights into the mechanisms of adaptive response to UVA radiation stress, and also comparing different hormesis treatments from low oxygen, low temperature, X-ray, irradiation, UV radiation points towards a unique response for each condition. And then leading us to understand that organisms adapt to produce gene products and in anticipation of environmental insults. So rather than reacting to stress by initiating transcription of genes, the organism upregulates the expression of said genes for the ongoing and future stress. There's plenty of work showing hormesis to anoxia, but little to no mechanism. And now we know some players. We know some gene products that are players in this process. So future directions and progressing us forward with not just this experiment, but other experiments involving environmental stressors. We can use hormesis to assist in finding an epigenetic clue in an organism's capacity to transfer stress to offspring. We also know that we can use the gene list from the last slide to show the insights into the mechanism of adaptation, resistance to environmental factors, and insects. And with that being said, I want to acknowledge all those who helped me along the way. I really appreciate the help from GC and allowing me the opportunity in his lab and to create a whole bunch of great things and travel to North Dakota and things like that to do an externship. I also want to thank my doctoral committee, Dr. GC, and Dr. Elba Serrano, Dr. Graciela Unguez, Dr. Henrietta Pachon, Dr. Peter Cook. And I want to say thank you to RISE and all those involved in funding research scientists like myself from underrepresented places to help us get funding to get some of the work done and do traveling and actually go to different conferences. I also want to say thank you to the New Mexico EMBRE Pilot Award for allowing the support in our transcriptomic study and the understanding of gene regulation and environmental stress. And then also like to point out my lab members who have gone to do great things, from Alyssa, Michael, Jackie, Nubia, and Lorena, who are doing great things in science right now and in their lives, and I hope the best in their future endeavors. And last but not least, I want to say thank you to my daughter, Hazel Neve Berry, for continually inspiring her father and pushing him to keep going with his dreams. And I hope she can see her father be a research scientist and a doctor now and possibly do the same thing or whatever she wants to do and put her heart to. Follow your dreams. And with that being said, I will take any questions that you have about the research or anything else. Thank you for attending.